Assalamualaikum and a very good day to everybody. So for group 2, we will be presenting about asexual reproduction strategies. So this is all of our team members, which consists of 4 people, and the work have been equally divided and everyone has contributed equally to the project. Next. So when we talk about reproduction in plants, we know that the topic is quite big and there's a wide range of strategies of reproduction to cover. So to make things easier, we are specified down into the topic of tissue culture. And to get more information about this field, we have conducted an interview with an expert on this part. About our interviewee, we have interviewed Dr. Muhammad Firdaus Ismail, and he is an expert in the field of agricultural botany. And this will help us to understand more and get more knowledge about our specific topic, which is tissue culture and reproduction.
Assalamualaikum warahmatullahi wabarakatuh. My name is AMIRAH WAJIHAH binti KAMARUDDIN. So today I'm going to talk about a method of a tissue culture. Okay, so as we know, tissue culture can be used in both plant and animal. Okay, but today I'm just going to mention about a tissue culture in plant. Okay, so without further ado, let's get started. Okay, so as we know, tissue culture is a technique in which a small fragment of a plant or its plant are introduced into an artificial nutrient medium which allow it to functioning or growth. Okay, so it is important to take note that the entire organism also used for a tissue culture, not only a small fragment of the plant. Okay, so the medium that can use for a tissue culture is a broth or agar. Okay, so agar is a nutrient dense gelling agent. Okay, so we know that plant tissue culture system are effective used in a commercial production as it can produce in a large scale. So there's a few type of a tissue culture. That is, first is a seed culture, embryo culture, and an organ culture. Okay, so for seed culture, orchid is commonly used for this type of a tissue culture. Okay, so for this method, eggs plant or tissue from the plant are obtained for an in vitro derived plant and introduced to an artificial environment where they can get a proliferate or increase in number. Okay, so plant material that will undergo the method need to be sterilized first to prevent the tissue damage and ensure optimal regeneration. Okay, so for the organ culture is a type of a tissue culture that involves isolating an organ for an in vitro growth. Okay, so an organ plant that can be used is a shoot, root, leaf, and a flower. Okay, so the last one is the step of a plant tissue culture. Okay, for the stage 1, the tissue need to be sterilized first to prevent any microorganism that will negatively affecting the process. So in this step, tissue will initiate to culture. Okay, and the second stage, that is the in vitro plant material is redivided and then introduced into the medium. Okay, so this medium is composed of a component for growth that include a nutrient. Okay, at the stage 3, the roots are formed and the hormones are required in order to induce rooting and consequently complete a platelet. That is all from me. Thank you.
Assalamualaikum and hi everyone. So I am MAZLEENA. I will continue with application of tissue culture. So firstly is rapid clonal propagation. A clone is a group of individuals or cells derived from a single parent's individual or cell through asexual reproduction. Therefore, all platelets regenerated from a callus or suspension culture generally have the same genotype and constitute a clone. So, second is somaclonal variation. Somaclonal variation is like a genetic variation. So basically, this variation has been used to develop several useful varieties. Third is transgenic plants. So, a gene that is transferred into an organism by genetic engineering is known as transgene. But an organism that contains and expresses a transgene is called as transgenic organism. The transgenes can be introduced into individual plant cells. The platelet can be regenerated from these cells. By this method, the platelet give rise to the highly valuable transgenic plants. 4. Induction and selection of mutation. So this is the flow. So firstly, mutation induction. Mutation induction is when the mutation are added to single cell liquid cultures. So next, it will isolation of induced mutation, where the useful mutants are selected for future breeding. So basically, it will test the mutation by tolerance it to stress like pollutants, toxics, salts, etc. Lastly, agronomic evaluation of mutants. The surviving healthy cells are taken to solid medium for raising resistance. So, lastly is the application by resistance to weedicides. So, firstly, the weedicides are added to culture initially in very small concentration. So, next, the dosage is increased in subsequent cultures until the desired level of resistance is obtained. Then, it will regenerate to form platelets and plants. That's all from me. Thank you.
The benefits of it is that you can produce plantlets, that is uniform. Uniform meaning that there is not much variation between the plantlet. Secondly is that plant tissue culture will allow you to produce mass number of plantlet at a time. Since tissue culture normally could produce faster plantlets compared to other conventional methods. In terms of producing plants that is having scarcity in raw material or propagation material. Of course, currently in our industry, grafting is one of the most common technique. So I think for conventional plants in Malaysia, you can use grafting, and then, but for big plantation, you have the skill and the financial strength that they can opt for tissue culture. The first thing is that in establishing or in coming out with the protocol itself, not all plant species will use or will have the same protocol to produce that. The biggest challenge for tissue culture activities is contamination. All right, tissue culture can easily be contaminated in the process. So first, you need to know what's your goals, and secondly, of course, the feasibility of technology at your hands. You need to consider that. Maybe it will save you cost or save you in a lot of other areas. Tissue culture can produce huge amount of plantlet in one time. But of course, tissue culture have its own hiccup, which is it require specific place, an established laboratory or a suitable sterile environment where you can conduct the procedures. So tissue culture can produce mass plantlets. Of course, it's a higher cost, but the economic return is actually quite good if you really plan use the right crop for that technique.
Assalamualaikum warahmatullahi wabarakatuh. My name is MIQDAD. So today I will talk about conclusion that we get from topic and from interview. So without further ado, let's go.
For the first one is conclusion from the topic that we studied. The second one is conclusion from the interview that we do. So that's all from me. Thanks for watching. Thanks for your attention. Have a nice day and Assalamualaikum.